Hello everyone, I am Saurabh and I welcome you back to the digital board series by Orchids Biosciences. This is the second video for Heredity and Evolution. If you haven't watched the first video yet, I have provided its link in the description below, so please do look for it and watch it. In this video, we are going to look into the following topics. We will start with knowing who is Gregor Johann Mendel, followed by his contributions. In this video, we will discuss about his experiments. But before understanding his experiments, first we will understand the various reasons for selecting the pea plants for his experiment. Gregor Johann Mendel selected the pea plants for his experiment, so we will understand the three major or the most important reasons for selecting pea plants. After that, we will discuss about the monohybrid cross in detail in this video, followed by the dihybrid cross in the next video. So in this video, we will discuss who's Gregor Johann Mendel, the reasons for selecting pea plants, followed by the monohybrid cross in detail. So this is an overview of what are we going to learn in this video. So starting with who is Gregor Johann Mendel. Gregor Johann Mendel is also known as the father of genetics. Gregor Johann Mendel was actually an Austrian monk who gave us the basic principles of heredity or the basic principles of inheritance of traits. We know heredity is basically the inheritance of traits from the parents to their offspring. So Gregor Johann Mendel gave us the basic principles of heredity and is also known as the father of genetics. So what did Gregor Johann Mendel experimented upon to come to the conclusions related to genetics? He worked upon the garden pea plants, whose scientific name is Pisum sativum. So before learning about his experiments, let us understand the various important reasons for selecting garden pea plants. Why did Mendel choose the garden pea plants? So first and a very important reason for selecting the garden pea plants by Mendel was that Mendel was able to observe or identify certain definite traits or definite characteristics, and these traits or characteristics in the pea plants were very clear or easily identifiable. So these definite traits or characteristics which Mendel observed in the pea plants were very easy to identify, and there were many definite or distinct characters which Mendel was able to see. So this was one of the very important and major reasons why Mendel chose the pea plant. For example, to understand this point in a much clearer way, the pea plants can be observed as tall pea plants or short pea plants. So we can see the tall character is very clear or the short character is very clear. So the traits, these are contrasting traits. For example, tall is contrasting to short. Tall is opposite to short in simple words. So tall character is contrasting to the short character. So he was able to identify such contrasting and very definite characters or traits. So both tall and short belong to the same character, that is height. So height can either be tall or it can also be short. So because he was able to identify such contrasting pair of characters, he chose the garden pea plant for his experiment. Along with height, he took another 6 pairs of contrasting characters. So totally he was able to identify 7 pairs of contrasting characters. Or traits. But why am I using the word pair? Because we can see it is occurring in pair, tall and short. So height has two possibilities. It can either be tall or it can also be short. We will be studying about this contrasting characters in detail in the further slides. So this was one of the reasons that he was able to identify certain definite traits or characteristics. The second reason was that the pea plants were annuals. So if they are annuals, which means their life cycle will be very short. So if their life cycle is very short, they can be grown very fast. So their growth will be very fast. And if their growth is very high, then he can observe a large number of generations in very less time. So this was one of the reasons. So because they're annual plants and their life cycle is very short, so due to short life cycle, what happens? Mendel will be able to observe large number of generation in his lifetime in a very small amount of time. So this was one of the another reason why he chose the pea plants for his experiment. One more reason is that the pea plants were self-pollinating in nature, which means naturally they were self-pollinating, and if we want we can also perform cross-pollination. So cross-pollination can also be performed. They are naturally self-pollinating. So this is by nature. So naturally they're self-pollinating, but we can also perform cross-pollination with the pea plant. So these are the most important reasons why he chose the pea plant for his experiment. Now when we are clear with the various reasons for selecting pea plants, let us understand these 7 pairs of contrasting characters which I mentioned here. We will be discussing all the characters in detail. So these were the 7 pairs of contrasting characters which Mendel chose. One by one we will discuss. Starting with one of the character, that is seed shape. So in seed shape, if we see, there will be 2 contrasting seed shape. They can either be round or they can also be wrinkled. So if we see, a gene is able to exist in 2 alternative form, alternative form 1, alternative form 2. So this alternative form of a gene is called as allele. So this is one allele, this is another allele. Out of the 2 alleles, one will be dominant and another one will be recessive. What is this dominant, recessive? Which one is dominant? We will be learning in the monohybrid cross in detail. So we have to remember that the genes exist in alternative forms which are called as allele. And among the 2 alleles, one will be dominant and one will be recessive. So here if we see, round character is dominant over wrinkled character. Similarly, every pair of contrasting character, there will be one dominant and one recessive. So round is one allele and wrinkled is another allele for the same gene, that is shape. So every gene will be existing here in 2 different forms. One will be dominant form and one will be recessive form. So let us quickly discuss the rest of the characters as well. After seed shape, another character is seed color or color of the seed. In seed color, yellow color is dominant over green color. So green is the recessive character. The dominant character is yellow seed color, whereas green seed color is the recessive character. So when yellow and green both are present, yellow will be dominant. When round and wrinkled both are present, round will be dominant. We will be discussing this in detail in monohybrid cross. Now we are only discussing what were the characters, what were the 7 pair of contrasting characters which Mendel chose. One was seed shape, one was seed color. Next one was color of the flower or flower color. In flower color, the violet color was dominant, whereas the recessive one was white color. So violet color flower was dominant over white colored flowers. So violet is the dominant character, whereas white is the recessive character. Or we can also say violet allele is dominant over white allele. Very simple. Next one is the shape of the pod or pod shape. In pod shape, if we see, one is inflated, which is dominant, and one is constricted, which is recessive. So in pod shape also we have 2 alternative forms. It can either be inflated, which is the dominant character, or it can also be constricted, which is the recessive character. Next one is the color of the pod or pod color. Here this one is recessive and this one is dominant. I have changed it because we have to remember with this color yellow. So here also I have put yellow and here also yellow. So yellow and green. So here yellow is recessive, whereas here yellow is dominant. So in seed color, yellow is dominant, green is recessive. Whereas in pod color, yellow is recessive and green is dominant. It's just opposite. So in color of the pod, yellow colored pods are recessive character, whereas green colored pods are dominant character. In all this, the left side is dominant, but here the left side is recessive. So in seed color, dominant is yellow and recessive is green, whereas in pod color, dominant is green and recessive is yellow. So this is reverse, seed color and pod color. Next one is the position of the flower, where it is placed. If the position of the flower is axial, this is axillary position. This is terminal position. So if the position of the flower is axial, that is dominant character. Whereas if it is at the terminal or the topmost position, that is a recessive character. So in axillary position, it is dominant, whereas in terminal position, it is recessive character. So axillary position of the flower is a dominant character over the terminal position of the flower. Terminal is nothing but the top or the apex, the top position of the plant. So these are the two contrasting characters. And what is the character here which we are studying? That is the position of the flower or flower position. Now one more character is pending, that is the height of the plant or plant height. In plant height, again two contrasting characters which we already discussed. This example we have already discussed, tall character and short character. Short character is also sometimes represented as dwarf character. So tall is dominant whereas short is recessive, or we can say tall allele is dominant over the short allele because they are the alternative form of the same gene. So this is tall, this is short. So these are the 7 pairs of contrasting characters. We have to remember green pod color is dominant over yellow pod color. So we have to remember this one and this one are opposite. So these are the 7 pairs of contrasting characters which she took. So each one forms a pair. So 1, 2, 3, 4, 5, 6, 7. So totally 7 pairs of contrasting characters. So now we are very clear with our basics. Now we can perform the monohybrid cross. So to perform the monohybrid cross, we have to take any one character. So for easy understanding, I will be explaining with the plant height as character. So now when we are clear with the basics, let us learn about the monohybrid cross. For easy understanding, we will be taking height of the plant as character. Now this height is available in 2 different forms. It can be either tall height or short height. This tall height is represented by capital T, whereas the short height is represented by small t. Why can't we write T here and S here? Because the capital one denotes the dominant character, small one denotes the recessive character. So if we write S here and T here, that means they do not belong to the same gene, but they belong to the same gene. Both belong to the gene, that is height. It is just the alternative forms of height. So since both belong to the same gene, we should use same alphabet. If we see capital T and small s, which means they do not belong to the same gene, or they are not the alternative form of the same gene. This is alternative form of one gene and this is alternative form of another gene. If we see capital S and small s, then we can say that these two belong to the same gene. So because they belong to the same gene, we have to use the same alphabet. So for tall height, we are using capital T. For short height, we are using small t. Now, when can a plant be tall or when can a plant be short? For a plant to be tall, there are 2 possibilities. So here there are 2 possibilities, and for short height there is only 1 possibility. So there are totally 3 possibilities which we will see. When can a plant be tall and when can a plant be short? If both the parents are tall, then the offspring will be tall. So this is the parent generation, this is the offspring or the next generation. If one parent is tall and one parent is dwarf, now out of tall and dwarf, which is the dominant one? Tall is dominant and dwarf is recessive. So the dominant character will be seen in the offspring. So offspring will be tall. So if both are tall, confirm will be tall. But if one is also tall, it will be tall in the next generation. So the only case where the offspring can be dwarf is when both the characters are dwarf, when both the parents are dwarf. It can also be represented as short. So when both the parents are short, the offspring will be short. Even if one parent is tall and the another parent is short or dwarf, it can be tall. So if both are tall, the offspring will be tall. If one is also tall, the offspring will be tall. Only if both are short or also called as dwarf, the offspring will be short. So these are the 3 cases or 3 conditions how the offspring will be. So this tall will give the gamete capital T. This tall will give the gamete capital T. So it will be capital T, capital T. This tall will give the gamete capital T. This dwarf or short will give the gamete small t. So this will be capital T, small t. Capital T, small t. So we can see there is a difference. This is capital T, capital T. This is capital T, small t. But this will also look tall. This will also look tall. Looking at the third condition. This is dwarf. So it will give the gamete small t. This is dwarf, so it will give the gamete small t. These 2 gametes will fuse to form another short or another dwarf plant, that is small t, small t. So this will be dwarf or can also be called as short and it will be small t, small t. So what is the difference between this tall and this tall? If we see here the zygote which is formed from the fusion of these 2 gametes, we will call it as homozygous tall. Homo means same, zygous means zygote. So the zygote is homozygous tall, which means both the alleles are of tall. Whereas here if we see, it is different. Two different alleles are there. One is tall, one is short. So we'll call it as heterozygous tall. If we look at the plant, this is tall, this is tall, this is short. So by looking at the plant or by seeing the physical appearance of the plant, whatever character we are telling is known as phenotype. So phenotype is nothing but physical appearance. Very easy to remember. P for phenotype, P for physical appearance. So phenotype can be tall or dwarf. So phenotype has two conditions. It can either be dwarf, also called as short, or it can be tall. So tall and short, there are only two possible phenotypes. But if you see the genetic composition, capital T, capital T, capital T, small t, small t, small t. So there are three possible allele combinations. Alleles are nothing but different forms of gene. So we can tell this as genotype. So genotype is nothing but the genetic composition. So genotype can be capital T, capital T, capital T, small t, and small t, small t. For these two, the phenotype will be tall. For this one, the phenotype will be short. So this is also very much required to understand the monohybrid cross. I will go over it again. If both the parents are tall, the gametes will also be having the tall allele. So the offspring will be tall. This is the first condition. If one of the parent is tall and one of the parent is short, since tall is the dominant character will be expressed. So the offspring will be tall. The only condition where the offspring can be short is only if both the parents are short. So only if both the parents are short, the offspring will be short. So only one condition is there where it can be short. Now when we look at their gametes, this is a tall plant. So gamete will be T, that is capital T. Here also will be capital T. So capital T will fuse to form two capital T's. So here it will be tall and it will be homozygous tall because the zygote will be having same type of allele, that is same, that is homo. Homo means same. So same type of allele will be present. Whereas in this case, if we see, one is tall character, another one is short character. So two different type of alleles will be incorporated into the zygote. So here it is heterozygous, which means two different. Hetero means different. And the final one, which is short. In short, there is no condition called homozygous short or heterozygous short. It is always homozygous. If it is heterozygous, it cannot be short. It has to be tall. So this is about how the plants are there. Either it is tall or short. Then we learned about the two terms, very important terms, phenotype and genotype. Phenotype is the physical appearance of the plant. Genotype is the genetic composition of the plant. Phenotype and genotype can be different. For example, here if we see, phenotype is tall, but the genotype can be capital T, small t also. But since capital T is dominant, that is tall is dominant character, it will be expressed out and the recessive character will be suppressed. So with all this information, we will be incorporating in the monohybrid cross. This monohybrid cross is basically a cross which involves only one character. So we are taking one character, that is height. Similarly, there is another cross called as dihybrid cross. Di means two. So dihybrid cross is a cross which involves two pairs of contrasting characters, whereas monohybrid cross will involve one pair of contrasting characters, because mono means one and di means two. Trihybrid means three pairs of contrasting characters we are going to study. So in this video, we will be learning about the monohybrid cross, in which only one pair of contrasting character is taken. Here, which is the contrasting character which we are taking? That is the height. We can take any of the 7 pairs of contrasting characters in study. So here we will start with height. So first, what Mendel did was, he took a pure tall plant and a pure short plant. What is the meaning of pure tall? Why am I not telling only tall? This is tall, but this is not a pure tall. This one is a pure tall plant. This one is a pure short plant. So he took one pure tall and one pure short plant. So let us perform the monohybrid cross. The parental generation or parent generation. In parent generation, he took one tall plant and short plant, which were pure tall and pure short. So the plant will be capital T, and here will be small t, small t. Now when this produces the gamete, it can either be capital T or it can only be capital T. There is no other possibility. If it was capital T, small t, then there was possibility that capital T, small t gametes were there. But this is not the case, because we are taking a pure tall plant and it is pure short plant. So small t, small t. So these are the gametes. of the parental generation. So this one is a parental generation, these are the gametes. Now what we are going to do is we are going to check the possibilities. What if this gamete fuses with this? What if this gamete fuses with this? So to understand the various possibilities, we use a square board which is called as Punnett square. So using this Punnett square, we will be able to understand the various combinations. So this is one parent, this is another parent. So one of the parents combination we will write here, and another parents combination we will write here. So first anyone we can take. For example, this two we are taking, so we will write here. Then this two we are taking, so we will write here. Now we are going to see the possibilities. What if this gamete combines with this gamete? So it will be capital T, small t. What if this gamete combines with this gamete? It will be capital T, small t. What if this gamete combines with this gamete? It will be capital T, small t. Now what if this gamete combines with this gamete? Capital T, small t. So if we see whichever gamete combines with whatever gamete, the final product is capital T, small t. This is called as the genotype. This is the genetic composition, that is capital T, small t. If capital T, small t is there, what will be the phenotype? We know capital T is the dominant character, so the phenotype will be a tall plant. So phenotypically, the phenotype will be a tall plant. That is a physical appearance. So the phenotype is tall and the genotype is capital T, small t. All are capital T, small t. There is no other option. So all the F1 plants which we observed, this is called as the F1 generation or first filial generation. The first generation produced by crossing the two parent plant were called as a first filial or F1 generation. This F1 generation plants were all tall plants with the genotype capital T, small t. With this we can understand the concept of which is dominant and which is recessive. What we can do is, if we don't know which is dominant, which is recessive, let us imagine there is a plant X and there is a plant Y. This is one character, this is one character. This is the parental generation. Now what we can do is we can cross these two plants. This is pure X, this is pure Y. So whatever is in the F1 generation coming, if in case X comes, then X is the dominant character. If Y comes, Y is the dominant character. Because we can see that in the F1 generation, only the dominant character is coming. The recessive character, that is short character, is not at all coming. So with this simple monohybrid cross experiment, with the first generation itself, we can figure out which is the dominant, which is the recessive character. For example, let us consider we took a plant which is having a yellow seed color and a plant which is having a green seed color. So when we perform this experiment, we will get to know that in F1 generation, we can see plants with only yellow colored seeds. So with this, we can come to a conclusion that yellow color is dominant over green color because the dominant character will be only expressed out. The recessive character will not be expressed. So with this, we can come to a conclusion that if we have two plants and both should be pure yellow and pure green, and if we cross these two plants, whatever is the F1 generation coming experimentally, that is only the dominant character. This is how we get to know it is dominant or recessive. Now which one didn't come? The green color seed didn't come. So green color is the recessive. So we can say yellow is dominant and green is recessive character. So this is the first step of monohybrid cross. What did we do? We took the parent generation, pure tall, pure short. We took the gametes from them, capital T, small t, small t. After this, we checked the possibility of combination of gametes and we came to a conclusion that all the possibility will give to only one type of plant, that is tall plant, and the genotype will be capital T, small t. After this, what he did was he crossed the two plants of F1 generation, which means any two plants you can take from the F1 generation and cross, because the phenotype, genotype, everything is same for all the plants. So when he crossed two plants of F1 generation, which means this is also tall, this is also tall, and we have to be very careful while writing the genotype. We have to write capital T, small t, capital T, small t. We should not make a mistake of writing capital T, because this is a hybrid, capital T, small t. It is not a pure tall plant. So this is called as F1 cross F1, that is also known as F1 selfing, which means we are self-crossing the F1 generation plants. So during F1 selfing, let us see what are the possible gametes. So in this, the possible gametes will be capital T, small t. Here will be capital T, small t. So these are the possible gametes. Now we will again use a Punnett square to identify and understand the various different combination of gametes to form their genotypes. So we can understand what a Punnett square is. Punnett square is basically a graphical representation of the possible genotypes. If we know the genotypes, easily we can identify the phenotypes. So using the Punnett square, we will see what are the combinations again. So here what we are going to do is, we are going to use this Punnett square to get the F2 generation. This is the second filial generation. The first one was the F1 generation. So in F2 generation, we are going to put these two belong to one parent. So here one, these two belong to one parent. So here one. So we are going to see all the possible combinations. If this gamete combines with this gamete, it gives capital T, capital T. This gamete combines with this gamete, it gives capital T, small t. This gamete combines with this gamete, it gives capital T, small t. This gamete combines with this gamete, it gives small t, small t. So now we will see what and all we got. We got capital T, we got capital T, small t. Two capital T, small t's we got, and one small t we got. This is tall, this is tall, this is tall, and this is short. So if we see, we got 3 tall plants and 1 short plant. These are possibilities. There will be many plants which will be grown. So the possibility ratio which we are getting here. So this is the genotype and this is the phenotype. So if we see genotype, this is one genotype, capital T, this is one genotype, capital T, small t, this is another genotype, small t, small t. So there are 3 possibilities of the genotypes, that is capital T, capital T, small t, small t, small t. How many capital T are there? 1. How many capital T, small t? 2. How many small t, 1. So 1:2:1 is the genotypic ratio. Next, if we see the phenotype, we can see there are 3 tall plants and 1 short plant. So the phenotypic ratio, that is tall is to short ratio, will be 3:1. Here 3 stands for tall and 1 is for short or dwarf. So the genotypic ratio is 1:2:1 and phenotypic ratio is 3:1. This is a very important question for your board examination. The genotypic ratio is 1:1, phenotypic ratio is 3:1. This is very repeatedly asked in board examination. So this is about the monohybrid cross. So if we see certain very simple conclusions which we can make is, there are 2 alternative forms of the same gene, and out of that one will be dominant, and the dominant one only will be visible in the F1 generation. Only when we cross the F1 generation we can find the recessive character in the F2 generation. So the recessive character will be hidden. It will not be expressed out until and unless the first generation plants were left to self-cross or they were self-pollinated. So these were the very basic conclusions from the monohybrid cross. The very important point to remember in this cross is the ratio which we obtained. Even if you forget the ratio, it just takes a couple of minutes to perform this cross and get the answer. So with this we come to the end of monohybrid cross. Now let us quickly recap whatever we have seen in this video. This is a very important topic as well. We started with knowing who's Gregor Johann Mendel. He is the father of genetics, who gave us the basic principles of heredity. After knowing who is Gregor Johann Mendel, we discovered the reasons for selecting the pea plants. One is the easily identifiable and distinct characters. We saw that there were 7 pairs of such characters, starting from seed shape, seed color, the height of the plant, position of the flower, etc, etc. We even discussed the 7 pairs of characters in detail. Out of that, the 7 pairs, each pair, if we take any one particular pair, there will be one dominant character in that and one recessive character in that. Whatever is the F1 generation, that is dominant. Whatever is not seen in the F1 generation is recessive. We can do this easily by monohybrid cross. The second reason was that it was having a very short life cycle. That is because they were annual plants. So because short life cycle, Mendel can observe a lot number of generations in his lifetime. After that, the next reason that we studied was they are self-pollinating in nature, which means naturally they are self-pollinating, and cross-pollination can also be performed. So these were the 3 important reasons that we discussed. After that, we discussed about the 7 pairs of contrasting characters, and finally we learned about the monohybrid cross. in monohybrid cross, before performing it, we learnt how a plant can be tall. Either the genotype is capital T small t, it can be tall. If the genotype is capital T, it will be tall. So these were the possibilities. If both are small t, then only the plant will be short. And these are called as the phenotypes. And this is known as the genotypes. For a plant to be tall, even if one of the parent is tall, the plant will be tall. But if only both the parents are short, then only the plant will be short. This also we learned. After that, we did the monohybrid cross, in which what we did is we took two pure plants as parental generation, capital T and small t. From their gametes, we got the Punnett square and finally the F1 generation. In F1 generation, we saw that all the plants were tall with the genotype of capital T small t. When we did F1 crossed with F1, capital T small t crossed with capital T small t, in F2 generations we found three possibilities of genotype: capital T, capital T small t, small t, and the ratio was 1:2:1. And we saw that these two are tall and this one is short. So tall was 3 and short was 1. So phenotypic ratio was 3:1, genotypic ratio was 1:2:1, and this ratio is very, very important. So this is about what we have seen in this video. Now let us quickly discuss the questions from the previous year. From this chapter only, there are very conceptual questions which form. Taking a simple 1-mark question, starting with it, a Mendelian experiment consisted of breeding pea plants bearing violet flowers. So the color of the flower we are taking here, with pea plants bearing white flowers. So we have two colors, violet. Character is flower color. One is violet color, another one is white color. What will be the result in the F1 progeny? So here if we say they have not given us which is the dominant, which is the recessive. So we have to clearly before the exam remember all the dominant character and recessive character and go. Then only we will be able to find the F1 progeny. So here violet color is dominant over white color flower, we have seen already. So violet is capital V. White can we write as small w? No, we cannot, because they belong to the same gene. So we have to write it as small v, not small w, so that it represents that both belong to the same gene. So here it is capital V, here it is small v. These are the plants. When we cross, what will be the F1 generation? Of course, it will be a heterozygous condition, capital V, small v, just like capital T small t was there. And the color will be the dominant character. Which is the dominant color? Violet color. So what will be the result in the F1 progeny? The result will be phenotype violet color flower and genotype capital V, small v. This is just similar to the capital T small t experiment. So if we know one thing very clearly and understood the concept, we can apply it anywhere. So if we write this alone, that is enough for the answer, because it is only a 1-mark question. This question can also be solved with the very basic knowledge of the fact that in F1 progeny always the dominant character will only come, and that also will be a heterozygous dominant. So if we know which is the dominant character among violet flowers and white flowers, we can easily answer this question. So in F1 progeny, since dominant character will come, it will be confirm a violet flower, and since it will be heterozygous, it will be capital V and small v. So with this fact also we can solve this question. No need to perform a cross, because it is a 1-mark question, we can directly give the answer there. Next, it is a very recent question. A green stemmed rose plant denoted by capital G, which means this is dominant. Since dominant is represented by capital G. So green color is the dominant character. Next, and a brown stemmed rose plant denoted by small g. So this is the recessive character. So with this we can understand that green color is dominant and brown color is recessive. So this is the basic information. Green colored stem plant and brown colored stem plant. Green is capital G, brown is small g. Now we will see what are they asking us in the question. It has been split up into two parts, A and B. First we will answer the first part, that is A. They are asking the color of the stem in the F1 progeny. We know that the color of the stem in the F1 progeny will be the dominant color, because in F1 progeny always the dominant character is seen. So without even performing the cross, we can say this, but we will perform the cross to understand it in a much better and a clearer manner. So we'll perform the cross. We have one green colored stem rose plant and a brown colored stem rose plant. Here green is the dominant character, so capital G, capital G. Brown is the recessive character, so small g, small g. This will be having gametes capital G, capital G. Here the gametes will be small g and small g. So this is about the parental generation. In the F1 generation, we know we will be getting capital G, small g as all the gametes, and the genotype will be green. So the first option we can answer very clearly. Color of the stem in the F1 progeny will be green colored stem. This is the first answer. Second one. Percentage of brown stem plants in the F2 progeny if F1 plants are self-pollinated. So they're telling we have to do the F1 selfing. F1 plants are self-pollinated. So if we do F1 selfing, F1 cross F1, we'll get G cross with GG. Same like capital T small t cross with capital T small t. It's just the same thing. The letters are different here. So they're asking us the percentage of brown stem plants, which means they're asking the percentage of the recessive character. Brown is the recessive character here, and they're asking us the percentage of brown stem plants. So we will cross this. So we know how we will get capital G is 2, capital G is 2, small g. We note the ratio 1 is to 2 is to 1. Just like the monohybrid cross of the plant height character. So they are asking us the percentage of the recessive character. We know this is the recessive character. So 1 out of 1 plus 2, 3, 3 plus 1, 4. So 1 out of 4 is the percentage. So if we want to convert it into percentage, into 100 we have to do. So 4 ones are 4, 4, 25 is 100. So 25%. So the percentage of brown plants in the F2 generation will be 25%. Now moving on to the third part of the question, they are asking what is the ratio of capital G and capital G small g in the F2 progeny. So they are asking only these two ratios to us, that is capital G is to capital G small g. We know the ratio 1 is to 2. We don't have to write here 1 is to 2 is to 1. Only what is asked we are supposed to write. If we write here 1 is to 2 is to 1, the answer will be wrong. So you have to carefully read what are they asking us. So this is the third part of the question. Moving on to the part B. Based on the findings of this cross, what conclusion can be drawn? Since it is a 3-mark question and we have already given so many answers, this doesn't have to be a very elaborate answer. We can give a very easy point for this one. We can tell that with this we can come to a conclusion that the green colored stem of the rose plant is dominant over the brown colored stem rose plant, and with this you can get 3 marks easily. Don't have to write very lengthy answers like any other chapter. This chapter is very conceptual. Only if you know the monohybrid cross clearly you can solve these questions very easily. Don't have to perform the cross again. So this brings us to the end of this video. In the next video we will be discussing about the dihybrid cross and sex determination as well. See you in the next video. Thank you so much for watching this video. If you like this video, please hit the like button, share it with your friends, and do subscribe to our YouTube channel.